So hello and welcome once again, dear friends, to the last point of this unit and the last point of regulation of gene expression, that is controlling gene expression at the translational level. When we speak about gene regulation, we are speaking about how much amount of a protein is to be produced, when it is to be produced, whether it is to be produced or not to be produced. All these decisions are taken by the cell. The interesting factor here, my dear friends, is to understand the fact that when we say like this, to take a decision, it becomes anthropogenic in nature, it becomes human in nature. A single celled bacteria does not have such type of functioning like what we have with our brains. So how does that cell decide these things which I just said? The quantity of a protein, whether that is to be produced or not produced. Not only that much, deciding the quantity, that is when to switch it off and when to switch it on. That is what regulation is all about. All these things become much more interesting when we study the gene as a operon. So just let's take a brief recap of the structure of a gene. I will not go into much details, but for what we have been seeing in this previous lectures, I will just try to summarize this in a few slides. What is the concept of a gene and operon? A gene is the functional unit of a DNA and its function is protein synthesis. Operon is that cluster of genes. Here in this picture you can see that this A, B, C, D, E, F, these are genes, and these are the enzymes which I am speaking about. These are the proteins which I am speaking about, which are needed for the metabolic activities to continue. Now these needs to be regulated, these needs to be controlled, whether switching it on and off or deciding the quantity. That is done by immediately adjacent genes. Now this red mark here is the boundary, a boundary which differentiates a gene into two different distinct parts. On the left you have a regulatory gene and on the right you have a structural gene. Structural genes are those genes which are responsible for the actual enzymes in action, that is the actual proteins which are needed for metabolism. And these regulatory genes, they may or may not produce proteins, but they are the site for acting of the proteins, and these are the points where the regulation takes place in operon system. Now when we are talking about operon, when we are talking about protein synthesis, we know very well mRNA has to be synthesized. The function is called as transcription, and that mRNA needs to be translated into proteins. We have already seen the basic mechanism of regulation, that is regulation at the transcriptional level. That is just the cell decides whether a mRNA has to be synthesized or not synthesized, the basic aspect. But you see, there is a time, or there is a time where mRNA is already synthesized and translational regulation does also takes place. We will see that in today's lecture. So you see, any gene on a chromosome, when I say a chromosome, a complete circular covalently closed structure of DNA, when we are speaking about that, this closed circular structure is made up of operons. Hundreds and thousands of operons may be there. What is the basic design of operon? Structural genes, regulatory genes, any gene operon. Regulatory genes, they may or may not produce a protein, and that protein, if it is produced, that is a repressor. It is not really taking part in these metabolically active structures. Okay, so the function of regulatory gene is to bind RNA polymerase, which it does at the promoter, produce a repressor, which will be done by any gene near or away from the cluster, and a site, that is operator for binding the repressor. Now you see the boundary line. These two genes are needed very near to each other. What is the function of structural genes? To produce a mRNA, to translate that mRNA into proteins, and that proteins which will be acting as enzymes. Now this is just a small picture. What is the larger picture? The DNA is covalently closed and circular. Okay, this is a circular DNA. And the genes which are there are genes all over the length of the chromosome. And each and every gene, like see now, you see the orientation as we move from this orientation and we come along, follow the cursor, follow the pointer. As we come along, see the orientation now. This is a promoter, this is an operator, and from exactly opposite direction another gene, some XYZ gene, whatever the gene. This is a polycistronic molecule. So this is ABCDE, this is ABCDE. It will be transcribed and translated. Okay, and you see the I gene here. I gene is the repressor gene, the actual gene which produces repressor, and repressor molecules which are proteins themselves, but they are not included in the structural genes. They are present in the regulatory genes. They may be produced by a gene which is near this P and O or away from P and O. Okay, this is very important. These I, you can see I is distributed throughout the length of the chromosome. It can be present anywhere on the chromosome, and the product of this I gene is a repressor. It produces a repressor, and any I gene product can control any operator. There are one single repressor controlling multiple operators. This is the larger picture. This is the higher level information which you should need to understand. So these regulatory genes, they may produce mRNA, they may translate these mRNA into proteins, and but not enzymes. These are not enzymes. They are always, this proteins is equal to here, that will be repressor but not enzymes. If they are from structural genes, they will be enzymes. So this regulatory gene product here, this regulatory gene product may produce a mRNA, but that product of translated mRNA is a repressor. So this is the bigger picture of a Operon. Now see, regulation of gene expression we have already seen at the transcription level. Today my point of interest in discussing is the translation level control. Now mRNA, once it is produced, mRNA is already produced, so there is a lot of energy invested there. Now there comes a situation in the cell that this mRNA must not be translated, otherwise it will be a wasteful synthesis. In this case, what does the cell do? The cell creates a translational repressor protein. As you can see, these are two different figures. See the first figure at the top here, a translational repressor. This is not the repressor same as that of the transcriptional repressor. This is somewhat different, a translational repressor, and this repressor blocks the movement of ribosomes. Okay, translational repressor protein which can bind to the mRNA and prevent the translation from starting itself. Doesn't allow the translation, so this is somewhat exaggerated figure here. Even this polypeptide is not initiated. Okay, and the second type of regulation which is usually found in prokaryotes is the repressor based control, which does not allow the ribosome to move forward, or a small complementary mRNA is produced which can bind to the mRNA. So it becomes mRNA hybrid. Okay, so this is a mRNA which is the transcript needed for translation, but a small complementary mRNA which can bind to the mRNA and prevent the translation from starting. Okay, so this translation doesn't start here. Why does it doesn't start? Because this ribosome movement is blocked by the coupling of the double-stranded mRNA. These two are the basic mechanisms of control. Regulation is control. Once this complementary mRNA is degraded, translation may start. Once this repressor is removed or degraded, translation may start. So regulation is putting it on and off. This is the level. But there are many other ways also in the cell by which the translation is controlled. We have seen that transcription, there is a formation of hairpin and loop-like structures on the mRNA which blocks the movement of ribosome. So that structures also there, as we saw in the tryptophan operon in our last lecture. So come, let us see the different types. There are 3 to 4 types of mechanisms by which these transcription and translation, they are regulated. But before that, I would like to just give you some more insights on the translational level of control for your notes. For taking down the notes, I would again request you to pause here and see the points. We are discussing a level of regulation, level of control, which is based upon the mechanism of translation. So protein production in the cell, it can be principally controlled at three different levels. We have seen that. That is at the level of transcription, my last lecture, through the availability of mRNA for translation, and then making changes in the mRNA or modifying the mRNA by using some terminator sequences or repressor, as we saw. That is through availability of mRNA for translation and modulation of the mRNA translation rate. Means, as we saw in the tryptophan operon, means if the substrate is less or more, then the ribosomal movement is more or less, and that determines the sequence of terminator hairpins to be formed or not formed. That is the second level. And third level is simply, once the mRNA is synthesized, if there is need for a translational control, degrade the mRNA. Once the formed mRNA is degraded, there is no translation. That is the third level of translational control, by eliminating the mRNA or degrading the mRNA. Here remember that though both transcription and mRNA degradation, this depends upon the movement of ribosomes or the availability of ribosomes, only translational level of protein synthesis regulation will be discussed here in the lecture. This whole lecture is dependent upon the translational control. And translational regulation, it refers to the control of levels of protein synthesized from its mRNA. Means once mRNA is synthesized, then from there onwards the control is being discussed in this lecture. Having said that, I would like to emphasize that in comparison to transcriptional regulation, the transcriptional regulation is more frequently followed in bacteria as compared to the translational regulation. Transcriptional regulation is preferred in prokaryotes. What is translational regulation? It refers to the control of the levels of protein synthesized from the mRNA, and this type is also functional in bacteria. When I say transcription is preferred because it is more energy efficient, translational control also is very much active because it allows the cell to overcome the stress, environmental stress, nutrient stress, pH stress, temperature stress, all the stress which is related with cell growth and metabolism. That is very efficiently controlled at the level of translation. So usually in prokaryotes, protein synthesis is controlled mainly at the level of transcription. 4 to 5 points you should remember here. Translational regulation of protein synthesis in prokaryotes is mainly through the control of initiation of transcription. Initiation takes place at the Shine-Dalgarno sequences. If you remember the SD sequences, SD sequences are the point or the space on the mRNA, part of the mRNA where the ribosome binds. The binding of the ribosome on the mRNA actually is initiated at the point of SD sequence. There itself not allowing to bind, not allowing the initiation of translation to occur. You know very well, protein synthesis is divided into three parts: initiation, elongation, termination. And termination is row dependent, row independent when it comes to transcription, and termination is dependent upon nonsense codon when it comes to translation. You know very well. So in translation, the initiation itself is controlled. When the initiation is not allowed, it is automatically regulated. So these circumstances of the individual mRNA or the cistron may or may not be started. That is one thing. Means all or no control. That is not allowing the mRNA or the polycistronic mRNA molecule to undergo translation. Initiation itself is blocked. So all or none, like that. When initiation is started, fourth point, when the initiation is started, rate of initiation is different for different mRNAs. Means the speed at which the mRNA is translated into proteins, that is the strength of the ribosomes to translate into proteins, is different for different mRNAs. So this is also a very important part of control. And mRNA in the prokaryotic cells, it is metabolically unstable. Remember that thing. mRNAs are not stable structures. And mRNA in bacteria, they undergo rapid synthesis and rapid degradation depending upon the environmental conditions and the cell requirements. So having said that, we should also understand that sometimes there is one type of mechanism in metabolism which we know very well, feedback inhibition. That also is regulation. Okay, accumulation of excess amounts of the translation products, that is enzymes or proteins, they are used to shut down the pathway by blocking the first step of the reaction, that is autoregulation or feedback inhibition. In this way, the fine tuning between level of the protein produced and the extent of the cell requirement, how much the cell is requiring that protein, that fine balance is maintained or achieved. So translational level of regulation of the protein synthesis is still of great significance in many special cases. But again, I would like to emphasize, in prokaryotes, the regulation of gene at the primary transcriptional level is usually exercised because of one simple thing. Maybe in evolution it has achieved that feat because it is energy efficient. Why to synthesize a mRNA when translation is not needed? Simple. But as I said again, translational level control also exists. So regulation of gene expression at translational level, we will see four different aspects. Regulation by attenuation, that is either by a hairpin structure or stem and loop structures. Regulation by translational type of repressors. These are not the repressors which are blocking the transcription, but translation. So translational level repressor proteins. Third is bacterial riboswitches, beautiful brilliant structures which form symmetric and asymmetric type of designs, internally complementation of mRNA, that is internal folding of mRNA, just like the tRNA structure. These mRNA, they fold internally and they block the movement of the ribosomes. The fourth is small mRNAs or small single-stranded mRNAs, or for that sake even DNA maybe, which bind to the complementary DNA and they block the movement of the ribosome again. So four different levels of regulation we will see. So the first is the tryptophan operon, which can be regulated by attenuation. Attenuation or attenuator structures, you know very well, these are the hairpin like structures. As you can see, these are the terminator regions which does not allow the movement of the ribosome on the mRNA. Up to some level, we have seen that these terminator sequences, they control the transcription also, process of transcription also. Okay, row independent type of termination is based purely upon these hairpin structures. Similarly, these hairpin structures can also block the movement of ribosomes. So regulation of bacterial operons in a coupled transcription and translation process is by these type of structures. Termination can be controlled by change in the RNA secondary structure, which determines the movement of the ribosome, and ribosomal movement also complementarily determines the formation of these structures. They both go hand in hand, as we saw in the leader sequences in the tryptophan mRNA. So tryptophan operon can be regulated by attenuation. An attenuator is an intrinsic folding of mRNA, intrinsic terminator, which is located between the promoter and the first gene of the tryptophan cluster. Attenuators can control the progression of RNA polymerase into the structural genes, as we saw in tryptophan gene. This inhibits the transcription as well as translation. So it is a dual control mechanism, but this can be a best example also of regulation, translational regulation too. Okay, so attenuator structures, they can be regulated by translation, and attenuated structures can regulate translation. Try to understand two complementary statements I am making here. Attenuator structures, that is the stem and loop structures, can regulate translation, means they can block the movement of ribosomes. They can block the movement of ribosomes, whereas this movement of ribosomes can regulate the formation of these attenuator structures. Recall, here you can see if this ribosome halts at the tryptophan codon in tryptophan operon, then 2 and 3 is formed. And if the ribosome advances into the second structure, then 3 and 4 are formed. So whether this 2 and 3 will be formed or 3 or 4 will be formed, that depends upon the ribosomal movement. So I repeat two statements. Attenuator structures can regulate the movement of ribosomes, and movement of ribosomes can control the formation of attenuator structures. All in all, both these events can be used by the bacteria to regulate the amount of protein synthesized. So the alternatives for RNA polymerase at the attenuator, it depends upon the location of the ribosome. We know that very well. So this is the second type of control, which is called as the attenuator, where the stem or stem and loop or the hairpin like structures are formed. Only a stem it is formed, it is called as a hairpin structure. If a stem and loop is formed, a hairpin and a loop is formed, it is called as a hairpin and loop or a stem and loop structure, as shown here. So hairpin structures of the mRNA can also regulate the transcription, can control translation is also. Here we are describing the blocking of translation by the formation of a hairpin and loop structure, which is called as the terminator structure or the attenuator structure. So this is the first type. The second type, as I have shown earlier in the slide, a translational repression mechanism in prokaryotes exists. Just like a repressor is involved in blocking the formation of mRNA, a repressor can block the movement of a ribosome, thereby inhibiting the formation of protein synthesis or formation of proteins. So this is the cluster gene product, which is a protein. These genes, this is control, that is the mRNA binds to a repressor. So these are called as translational repressors, and they are very much active in the prokaryotes. You see this figure, this is taken from a research paper, but it is very simple. Two levels, one in the box and one outside the box, figures are shown. One in the box, dotted box, it shows the normal procedure, that is active mRNA. This is red structure is mRNA, which binds to the 30S subunit. Now we are talking about initiation of protein synthesis, initiation of the translation process. So what is the normal process here? Normal process is the active mRNA. It binds, GTPs are required, energy is required, 30S subunit it binds, then it forms an initiation complex. Recall the lecture on process of translation, the structure of ribosomes. You will understand that here F-met tRNA, this F-met tRNA comes, it binds a 30S subunit, then now couples to the 50S subunit, which is not shown here, a 50S subunit, and the translation starts. Now what happens in a control? Now let us imagine mRNA has been produced, but the cell suddenly encounters a situation. There is a situation where the protein is not needed. So what does the cell do? The cell instantly inactivates this mRNA. How does it inactivate the mRNA? By forming a repressor. So here the example in this bacteria E. coli is shown as S4 repressor. There is a S4 repressor which binds to the mRNA at the site of binding of the 30S and the 50S subunit. So this actually binds and inactivates the mRNA. So this S4, this can be an example of sort of a competitive inhibition. So here this S4 binds and allows the 30S subunit and the 50S subunit to compete and not allowing them to bind at the site. So entrapped inactive complex, as you can see here, even though it is bound, it is a type of competitive inhibition where a repressor is bound, 30S subunit is bound, F-met is bound, but these complex doesn't allow the 50S subunit to bind. So repression of this operon is by entrapment mechanism here. Two distinct forms of mRNA are involved in the translational regulation. This is an entrap mechanism by use of repressors. Two distinct forms of mRNA are involved in this translational regulation. One is the active fraction of the mRNA, which participates through the normal translational initiation pathways, which we have seen here. The second is the inactive mRNA fraction which specifically is bound by a S4 repressor and 30s subunit and forms a entrapped ternary complex. This is called as an entrapped ternary complex. At normal temperatures, at physiological temperatures, this becomes inactive and active mRNA conformations, they are not allowed. Okay, so this is how the translational repressors are active. In many bacteria, just raising the temperature, if a temperature is raised, the repressor is removed. So there are different mechanisms of removal of this repressor also. Okay, that is all dependent upon the environment, the conditions, the nutrients in which this bacteria is growing. So this is the second type of regulation or the control, which is called as translational repressor control, or you can say as entrapment method by repressor for controlling the translation. Okay, the third is bacterial riboswitches. Bacterial riboswitches, they function as regulatory elements in the mRNAs. What is a riboswitch? I will just skip this for the time being. Go to the next slide, show you a riboswitch structure, and then we will come back to this point discussion. Okay, point wise discussion. So see this is a mRNA, this green region is mRNA, and this is one single strand of mRNA. So what is happening? These are internally folded. There are many stem and loop structures in one single mRNA, and in the center there is a small protein or some other, it may be a metal, it may be protein, which is called as a ligand. And this ligand or this regulatory protein is responsible for this structure formation. These structures are called as riboswitches. In the internal center of mRNA, so you see there is a long length of mRNA, there is a ligand molecule which comes, and suddenly this mRNA folds around the regulatory protein, forming such beautiful structure. Here you see a symmetrical structure. It can be asymmetrical, irregular also. Okay, a regulatory protein, it binds to the riboswitch. This is called as a riboswitch, and it stabilizes the riboswitch and does not allow the ribosome binding site. You see this dark green center is the ribosome binding site, and this is complementarily bind to the another region of the same mRNA length. And so this gene is now this is the structural gene. This gene is not translated into protein. So this structure is called as a riboswitch. We will see, go back to the previous slide and see some interesting aspects of this riboswitch. Riboswitch, as I just showed, is a regulatory sequence of a mRNA molecule where molecules can bind and affect the gene expression by influencing the formation of a secondary structure in the mRNA. A secondary structure in the mRNA is formed and it influences the binding of the ribosome. No binding of the ribosome takes place due to that structure which is called as a riboswitch. Riboswitches are gene control elements which directly bind to the specific ligands, which is called as the repressor protein, and thus it regulates the gene expression without the need for any extra proteins. Okay, there are no extra proteins needed, and that regulation is carried out. These riboswitches, I would like to emphasize, are found in all three domains of life, including bacteria, archaea, eukaryota, everywhere. Riboswitches are mostly spread in bacteria and archaea, and more than 28 different types of riboswitch classes, they are discovered in bacteria. Bacterial riboswitches, they regulate gene expression by 4 distinct mechanisms. There are different ways of regulating by these structures. The details you can study, those who are interested, by taking out the information from the internet. But I just want to show you here, I will not go into these 4 different mechanisms. Only you have to understand that riboswitches, they regulate not by one particular structure and one particular mechanism. There are different mechanisms in different species of bacteria. They regulate the expression of limited number of genes. Means this is not a very common structure. Okay, very unusual structures. However, most of these genes which are responsible for the synthesis of essential metabolites, hormones, proteins, enzymes, they are regulated, without which the cell cannot function, without which the cell will die. these are regulated. So these are the bacterial riboswitches. As you can see again, secondary structures formed due to internal folding of a single length of a single-stranded mRNA molecule. Single-stranded mRNA molecule folding to form such beautiful structures called as riboswitches. So what happens when this riboswitch structure is formed? The ribosome binding site, this is the Shine Dalgarno sequences, SD sequences, which allows the ribosome to bind, and this is masked. This is internally converted into a temporary double-stranded mRNA structure, so ribosome can't bind. So no translation takes place. So automatically protein synthesis is regulated. When the regulatory protein inside here is absent, without the regulatory protein, the riboswitch, it takes a shape of alternative structure, another structure, and what happens, it just expands, it removes, and you can see the ribosome binding site is free. Ribosome will bind, that is without the ligand or without the repressor protein, and translation takes place. So this is the beauty of the riboswitches. Okay, so there are more than 28 different types of riboswitches discovered in bacteria. I have shown you here one structure which is symmetrical in this slide. Now you see the next, there are A, B, C, D different types of switches which control the ribosome binding site, which is called as RBS. These are allowed or not allowed, not allowing the ribosome to bind. Okay, so different type of structures are there. So again, this figure taken from a research paper is just to give you idea of the different types of structures formed, and there are certain structures which can, small structures like the complementary mRNA, which is the next point of discussion, also being formed. So these are various types of riboswitches which are controlled either by thermosensor or by activation by another mRNA or some proteins. It is a beautiful mechanism. A whole new world of translational regulation can be studied in a totally different lecture. So that's up to you to explore. That is the third type of translational control, that is riboswitches. Now the part which I would like to show here is that the translational initiation repression is the most important step which is followed by bacteria. That is blocking the 30S binding site, blocking the excess of 30S site by preventing the 50S to bind by repression. So this is one area, and the second area is degradation of mRNA. So either the initiation is blocked or the mRNA is completely degraded. The last type of translational control which we are going to discuss here is the complementary mRNA mediated translational regulation. So you can see a short mRNA is synthesized. Two different structures I am showing you. Come to this A structure here, follow the laser pointer. This green line here is the mRNA. Normal mRNA which is synthesized by transcription. Transcription is going on. But suddenly there is a small mRNA which can be present in the cytoplasm. The somewhat similar example I would like to give you, like the RNA primer which are used in DNA replication. Just like a small primer, RNA primer, which is complementary to the DNA, the open DNA, double-stranded DNA during replication. When it opens up, there is a small RNA molecule which initiates the replication. Just like that, a small RNA molecule from, now that is a different point of discussion, whether that is synthesized specifically or it is already present. It may be already present as a part of evolution. So I am not going to discuss that. Here you have to understand there are small mRNAs which by two mechanisms can block the movement of ribosome, can block translation. One is end-to-end ligation, another is complementary base match. End-to-end ligation is, as you can see here, there is a cut in the mRNA, and end-to-end these small RNAs, they get inserted into the normal mRNA, this orange mRNA, which you can see here. I would like to zoom in. Let us see if I can zoom it in. let us zoom and see this structure. So now I hope you can see the structure here. The green is the mRNA. This green length of segment is the normal mRNA. So what is happening with this orange? This is an extra material which is not transcribed by the regular gene expression product. It is not a regular gene expression product. It is bought from the cytoplasm and it is inserted in random in between the normal mRNA, and a terminator hairpin may be formed and it blocks the translation. One mechanism. Or instead of getting inserted, you can see the second example here, B, example B. What happens? This complementary mRNA, it just forms a temporary double-stranded mRNA structure. This is mRNA and mRNA, and this also blocks the movement of the ribosome. So translation is blocked. So these are the two instances, that is end-to-end ligation and complementary base match, which blocks the translation. Why I am always talking about blocking of translation? That is what we are trying to discuss, how the gene expression can be regulated at the translational level. So if you have to regulate it at the translational level, then the movement of the ribosome has to be blocked. So RNA mediated translational regulation is the main point of discussion here. Okay, so this is the fourth type of regulation, where small complementary mRNAs, they mediate the blocking of these ribosomes either by end-to-end ligating and forming terminator structures, or simply directly binding at some region which is having a complementation, means A, T, G, C pairing takes place here, and this also blocks the movement of ribosomes. So these are the various methods by which the gene expression can be regulated. So a brief recap again. Regulation of the gene expression takes place at the translational level. This can be done by various mechanisms. Regulation by attenuation, that is hairpin and loop structure. Regulation by repressors. Regulation by complementary mRNA sequences. So we come to the end of this lecture, and my next lecture will be on the next unit, which deals with the molecular biology techniques and applications. We will be mostly discussing about the recombinant DNA technology. We will be discussing about the basic aspects of genetic engineering, which mostly deals with what you see in the picture here. This is of course an exaggerated picture. It doesn't take place like what is shown here, that is cutting and pasting a DNA. When I say cut, a DNA is cut and paste. So you cannot hold the DNA, you cannot put scissors and glue and paste like this. This is just what you can say exaggerated expression for your understanding, that we can cut the DNA, we can paste the DNA, we can create chimeras, we can create newer molecules which can act and express the genes as we want. Think about it. Means modulating the gene expression, creating designer molecules, acting God maybe, as some of the scientists they say, geneticists, they are trying to act God. Now there is a lot of ethical issues involved in this molecular biology techniques all over the world, but our point of discussion will not see that perspective. We will see the wonderful techniques which are available in molecular biology by which we can create recombinant DNA molecules. So that will be the part of my next discussion, which you can find it in the next playlist. I am going to change the playlist for this. For this transcription level control, see the links below. There are some beautiful links which I have given for you to take down the notes. Again, repeat, see again and again, pause wherever necessary, take down your notes, and I emphasize I am always free for discussion. Ask questions whenever and wherever, by whichever mode you want to ask, and we can have a healthy discussion on these beautiful techniques in genetics which have created scientists to a level where now they can literally control the phenotypic and the genetic character expression of any living molecule on this earth. So till that time, all the best for your studies. Good luck.